Welcome friends and welcome to my channel Sequential Biology. We were talking about viruses. Today we will discuss about structure of tobacco mosaic virus. And you know tobacco mosaic virus is first discovered virus. Before going to discussion about a structure of tobacco mosaic virus, first we see that how this tobacco mosaic virus was discovered. Martinus Beijerinck of the Netherlands investigated the property of agent that causes tobacco mosaic disease, and first tobacco mosaic disease was called as spot disease. Then what is tobacco mosaic disease? Actually, when tobacco mosaic virus attack any tobacco plant, different spot can be seen in their leaf. The leaf usually very deep green color, but different spot or the yellow spot or light green spot appear in their leaf. So this was formerly called as spot disease, and later it was known as tobacco mosaic disease. Okay. Now, how this tobacco mosaic virus was discovered? When an affected plant, so this is an affected plant, and the parts of this affected plant first extracted and make sap. Okay, so this extracted, suppose this is leaf and the leaf portion are extracted, and then this was added with water, and this will form a sap. Okay, so this was added with water and this form a sap, and this extracted sap then passed through a bacterial porcelain filter. What is this bacterial porcelain filter? Bacterial porcelain filter contains different cavity, and in this cavity the bacteria can trap in this cavity because bacteria size is very much large. And when this extracted sap passed through bacterial porcelain filter, then the bacteria trapped in this filter, and only this particle actually passes, and which is smaller than this bacteria. Suppose this extracted sap contain viruses and the bacteria, and when this sap passes through this bacterial porcelain filter, the bacteria trap in this filter, and only the smaller particles than bacteria passes through this filter. Okay, and when these materials, and which is separated, these materials, that is the smallest and the tiny material, this sap is rubbed into a healthy plant, the healthy plant becomes affected. It is clear that these viruses are much smaller than this bacteria. Okay, now according to Beijerinck, a replicating particle much smaller and the simpler than bacteria so passes through bacterial porcelain filter. And he is first scientist to voice the concept of virus. So when filtered sap was rubbed on healthy plants, they become infected. Another scientist, Wendell Stanley, American scientist Wendell Stanley in 1935 crystallized this particle and now known as tobacco mosaic virus. This is very, very important in examination point of view. Who first crystallized tobacco mosaic virus? Wendell Stanley first crystallized this tobacco mosaic virus. Now, what are the symptoms of this tobacco mosaic disease? The light green coloration between the veins of young leaf appear. So these are the veins, and in between these veins the light green coloration can be seen, and also followed by mosaic development. Mosaic means net-like light green coloration can be seen, and curling of leaf, and the distortion of the leaf means this is a normal shape of a leaf, and curling means the leaf becomes curled. Okay, or the distortion or the shape will be changed, and this is called distortion of leaf. Now, tobacco mosaic virus was discovered by Wbssc in 1892. Then, who first voiced about virus? Beijerinck first voiced about virus, and he first told that, but before discovery of the viruses, the scientists believed that any of the diseases are usually caused by bacteria. Actually, they have no idea about viruses, but later Beijerinck first told that there are also smaller particle than this bacteria, and which may causes different type of disease in any organism. So he is the first scientist who voiced about virus. And Stanley first crystallized tobacco mosaic virus. But the tobacco mosaic virus was discovered by Dmitry Wbssc in 1892. W. Stanley in 1935 crystallized it and found the crystals are highly infectious. So tobacco mosaic virus crystals are very much infectious, and the Franklin described tobacco mosaic virus structure. Then Franklin first described the tobacco mosaic virus structure. And you know, any virus contains a capsid layer, so tobacco mosaic virus also contain capsid layer, and the unit of this capsid are usually capsomere, and the capsomere are grape shaped. So these are grape shaped structure. and the capsomere are grape-shaped structure, and this capsid is made up of many capsomere. These are the capsomere. Unit of capsid are capsomere, and this is usually this capsomere in tobacco mosaic virus are grape-shaped structure. Tobacco mosaic virus is a hollow rod-shaped virus. In the central core there are presents a hollow structure, and this is rod-shaped virus. Mosaic virus is hollow rod-shaped virus with a rough surface and looks like a bunch of banana. So this is looks like a bunch of banana. Each rod is 300 nanometer. So the length of each rod, suppose this is a tobacco mosaic virus, this is a tobacco mosaic virus, this is a tobacco mosaic virus. Each tobacco mosaic virus are rod-shaped structure, and the length of this rod is 300 nanometer, and their diameter is 70 nanometer in diameter, and this is obviously consisting of the two part. One is the protein coat or the capsid, and another is their nucleic acid. Then this is a rod-shaped structure, and the length of this rod is 300 nanometer, and also their diameter is 15 nanometer. Okay, so this is diameter and this is their length or the size of the 300 nanometer. And also there are presents a single-stranded RNA. So tobacco mosaic virus is a single-stranded RNA containing viruses, and their capsid is made up of subunit which is known as the capsomere, and this capsomere is usually made up of polysaccharide. So this is a polysaccharide structure, and capsid consisting of 2130 protein subunit, okay, or of identical size, and each subunit is made up of single polypeptide chain with 158 amino acid. Suppose this is a capsomere, and this is a capsomere, this is capsomere, this is capsomere, and there are present 2130 capsomere or protein subunit. So this protein subunit are nothing but this is capsomere, and there are present 2230 capsomere, and since each capsomere is a protein, so it must be consisting of different type of amino acid, and in each capsomere there are present 150 amino acid. Okay, each subunit is made up of single polypeptide chain with 158 amino acid, and the protein subunits are called as capsomere. Okay, now the nucleic acid is single-stranded RNA. Actually, the viruses show their genetic material in different variety. Some viruses may be double-stranded DNA containing virus, some may be RNA single-stranded DNA containing viruses. Some viruses may be double-stranded RNA containing viruses, some may be single-stranded RNA containing viruses. Then this tobacco mosaic virus is single-stranded RNA containing viruses which is spirally coiled to form a helix. Actually, these RNA are spirally coiled. Okay, so this RNA is not straight, this is not straight, but this is spirally coiled helix, and consisting of 6500 nucleotide. Actually, there are present a single RNA, and this RNA, you know, the RNA consisting of different type of nucleotides, and there are present 6500 nucleotide. And if we analyze their structure, this is, suppose this is a single-stranded RNA, and this single-stranded RNA is made up of 6500 nucleotide. These are nucleotide, and the 6500 nucleotide are there in their RNA. Okay, now the capsomere are closely packed and arranged helically around RNA helix, and this capsid actually covered this RNA helix. This is RNA helix, and the capsid covered this RNA helix, and thereby it forms a hollow cylinder about 4 nanometer in diameter. Since tobacco mosaic virus is a hollow a rod-shaped structure, and at the core there are present a hollow cylinder, and the diameter of this hollow structure is 4 nanometer in diameter. Okay, then this is 4 nanometer in diameter. The capsomere are closely packed and arranged helically around helix, and thereby it forms a hollow cylinder about 4 nanometer in diameter, which is present throughout rod. And RNA is deeply embedded in protein subunit. Then the RNA actually not fully present inside this core. So this is deeply embedded in protein subunit 5 nanometer from outside, so does not occupy the hole. Suppose this is a hole, this is the core structure of this tobacco mosaic virus, and outside of this hole the capsid layer are present, and the RNA covered, and this RNA is covered by this capsid, and this RNA not fully present inside this hollow core. This is very, very important. Remember this RNA. So RNA is deeply embedded in the protein subunit. Then this is protein subunit. Suppose this is protein subunit and the RNA is fully embedded in this protein subunit. So does not occupy the whole. The amount of the protein in rod is 95%. The amount of protein in rod is 95% and rest 5% is RNA. So maximum portion of this tobacco mosaic virus is made up of protein and only 5% is RNA. A single capsomere is linked with 3 nucleotide of RNA. A single capsomere linked with 3 nucleotide of RNA. So a single capsomere, suppose this is a single capsomere and this single capsomere linked with 3 nucleotide of RNA. This is actually the structure of tobacco mosaic virus, and hope you have understand the structure of tobacco mosaic virus. So thank you, and that's it.